A novel role for xanopsin, stimulation of food intake. This research was prompted by the initial question, will xanopsin produce the same anorexic genetic factors as xenin does on the digestive tract of newly hatched chicken models, which led to the hypothesis, since xanopsin has similar amino acid sequence structure as xenin, it will also be associated with reduction of food intake. It was previously found that xenin shifts motivation to exploration of the environment instead of food intake, while also showing an increase in activity in the ventromedial hypothalamus. Due to this knowledge, the hypothesis of this experiment was crafted based on the fact that xanopsin and xenin are structurally similar. Before I begin, I will briefly go over some background knowledge needed to understand this experiment. The body's system for regulating food intake is coordinated by the hypothalamus, which is located under the midline of the brain behind the eyes. This experiment will mostly involve experimentation of the lateral hypothalamus. The lateral hypothalamus is known for regulating metabolism and food intake. Stimulation of the lateral hypothalamus induces eating, while destruction results in starvation. Long-term regulation of food ingestions involves neuropeptides, those stimulate or inhibit feeding. Within the hypothalamus are nerve cells that, when activated, produce a sensation of hunger. They do so by producing two proteins that cause hunger, neuropeptide Y and AGRP. Additionally, another set of nerves contributes to the opposite effect on hunger. These nerves produce two different proteins that inhibit hunger, cocaine and amphetamine regulated transcript, or CART, and melanocyte-stimulating hormone. Circulating NPY originates predominantly from the sympaadrenomedullary nervous system. It has a vasoconstrictive and mitogenetic effect on blood vessels and seems to be involved in blood pressure regulation and angiogenesis. Stimulation of the NPY-ergic pathway by exercise, fasting, and energy loss is followed by increased appetite and food intake and increased parasympathetic activity, but suppression of sympathetic activity and energy expenditure. The end result of this process is an increase in energy storage. These hormonal circuits impact the sensations of hunger exerted via hypothalamic neuroendocrine pathways. The effect of these peptide hormone receptor interactions are the release of either the orogenic neuropeptides NPY and AGRP or the anorexic genetic peptides CART and the POMC-derived peptide. These neuropeptides from the ARC travel across axons to secondary neurons and other areas of the hypothalamus, such as the paraventricular nucleus. The ultimate effect of these signaling cascades are changes in the sensation of hunger and ultimately feeding behavior. The materials used for this experiment were chicks taken on the morning of hatch from Cobb 500 and Herbert broiler chicks from breeders 30 to 40 weeks of age. The methods of this experiment include freehand intracerebroventricular injection procedure, where the head of the chick was briefly inserted into a restraining device that left the cranium exposed and allowed for freehand injection. After data collection, the chick was decapitated and its head section along the frontal plane to determine the site of injection. Any chick without dye present in the lateral ventricular system was eliminated from analysis. The next method used was the cannulation procedure of 16 days post-hatch chicks. Injection was guided with stereotaxic coordinates, and if water intake increased, we knew that the location was right. The 8-part experiment began with Experiment 1, which studied the effect on food and water intake in 4 days post-hatch chicks after a 3-hour fast. Food and water consumption were then measured every 30 minutes for the 180 minutes post-injection. Experiment 2 and 3 looked at the effect on food and water intake after 4 days of free feeding. Experiment 2 had the same injection procedure as Experiment 1, while Experiment 3 included higher doses. Experiment 4 was designed to study CFOS immunohistochemistry. The chicks were randomly assigned to receive either 0 or 1 nanomoles of xanopsin via freehand IVC injection. Food was withheld to prevent CFOS immunoreactivity associated with food consumption. The brains were removed from the skulls and post-fixed for 1 hour in solution until they sank. They were then studied to determine the amount of CFOS immunoreactive cells within each receptive nucleus, counted by individual blind to treatment. Experiment 5 was to study the effect of xanopsin in 16 days post-hatch. The chicks were randomly assigned to receive either 0.5 microliters or of 0.5 0.3 or 1 nanomoles of xanopsin in a Latin square design, meaning treatment was assigned at random. Then their food and water intake was monitored every 15 minutes for 180 minutes post-injection. Experiment 6, 4 chicks were assigned randomly per treatment to receive either 0 or 3 nanomoles of xanopsin in a crossover design in order to study the effect of xanopsin microinjection into the lateral hypothalamus in 16 days post-hatch. Experiment 7, chicks were again randomly assigned and received either 0 or 1 nanomoles of xanopsin via freehand injection. 1 hour post-injection, the whole hypothalamus was collected in RNA-laden and homogenized in order to study the whole hypothalamus and expression of appetite-associated mRNA. Lastly, experiment 8 was designed to observe behavior. Prior to the observational period, the chicks were kept in individual cages with only auditory contact in order to reduce isolation stress during individual observation. Then the chicks were moved into individual clear arenas with access to food and water, where they were observed via 30 minutes of recording from 3 different camera angles. Now for the results. In experiment 1, xanopsin did not exert an effect on food intake or water intake at any time point measured. In experiment 2, those injected with 3 nanomoles of xanopsin increased food intake from 30 minutes to 150 minutes following injection. The water intake was not influenced during the 180-minute observation period. Experiment 3, the chicks injected with 1 and 3 nanomoles of xanopsin increased food intake through the 180 minutes following injection, while the higher injected 9 nanomole decreased water intake through 60 minutes following injection. Experiment 4, those injected with xanopsin had a 150% increase of CFOS immunoreactivity in the lateral hypothalamus, while the other hypothalamic nuclei measured were not affected. The top leftmost graph shows the mean and standard errors of the number of reactive cells. The next figure highlights the photomicrographs of the CFOS immunoreactivity within the reactive lateral hypothalamus and the non-reactive ventral medial hypothalamus. In experiment 5, chicks injected with 0.3 and 1 nanomole of xanopsin increased food intake from 30 to 45 minutes. In experiment 6, food intake increased following the microinjection of xanopsin into the lateral hypothalamus. In experiment 7, POMC mRNA expression increased in the whole hypothalamus for chicks injected with xanopsin. However, the abundance of AGRP and NPY mRNA was not affected. In experiment 8, the chicks that were injected with xanopsin showed different count type behaviors shown in Table 1 at 30 minutes following injection, while xanopsin-treated chicks increased feeding peaks between 20 to 60 minutes. Time-typed behaviors were also affected, shown in Table 2. Interpretation of the results from Experiment 1 was that if xanopsin was affecting food intake, it was increasing it. This led us to design the Experiment 2, which demonstrated that the central injection of the 3 nanomoles of xanopsin stimulated food intake, leading us to consider higher doses in Experiment 3 at 9 nanomoles of xanopsin, which surprisingly failed to increase food intake, meaning that the results were quadratic. Therefore, xanopsin injection is capable of stimulating an orexigenic effect only within narrow dose range. Experiment 4 led us to determine which hypothalamic nucleus was associated with the xanopsin orexigenic effect, and we found that the only reactive nucleus was the lateral hypothalamus, which is consistent with the documented role of hunger perception. Experiment 5 demonstrated that the 16-day-old chicks persisted a response lasting 45 minutes in comparison to the 4-day-old chicks' response of 150 minutes. Experiment 6 further demonstrated that food intake increased to support that the receptors associated with the orexigenic response are in the lateral hypothalamus. In Experiment 7, the chicks injected with xanopsin had increased POMC MRI expression. Because POMC may be cleaved into numerous biologically active neuropeptides, it is unclear if POMC itself or if one of its derivatives are affected. It should also be noted that xanopsin mechanism may involve extra hypothalamic regions stemming from the lateral hypothalamus projections to ultimately stimulate food intake. Behavior analysis in Experiment 8 was conducted to determine if behaviors unrelated to appetite were affected. Chicks injected with xanopsin had increased feeding peaks, which makes sense with the increased food intake demonstrated in the previous experiments. All other behaviors that were affected by xanopsin, deep rest, and sitting were decreased. This may imply that xanopsin has a primary effect on increased food intake, which causes the other behaviors to be decreased secondarily. In conclusion, we have provided evidence that xanopsin exerts an orexigenic response in chicks. This response is dose-dependent, as higher doses may induce malaise. The lateral hypothalamus appears to be the hypothalamic nucleus mediating the orogenetic response, and POMC or its derivatives may also be associated. Further research on zonopsin on larger animals with varying doses, as well as research of xenin and how it works to suppress hunger in contrast to zonopsin, along with further investigation of POMC mRNA expression, should be conducted. The only critique of this experiment is that the chicks are structurally related, and that may imply that they are acting via a common receptor, which could be the reason why zonopsin and xenin both affect food intake. And that concludes this experiment. Thank you.